Welcome back to O-Level Oasis. We are studying O-Level Biology 5090, and today, after completing all 12 chapters, we will begin our past paper practice for Chapter 12, Disease and Immunity. So let's begin. Alright, let's look at our first question. This one is a matching type question. The statement says, transmissible diseases are caused by pathogens. Now, your task is to connect each disease to the correct type of pathogen that causes it. So, we have 3 diseases listed here, AIDS, cholera, and malaria. And on the other side, we have the types of pathogens, virus, bacterium, and protoctist. Let's go through them one by one. First, AIDS. We know AIDS is caused by the HIV virus, so here we match AIDS with virus. Next, cholera. Cholera is an infection of the intestines caused by the bacterium Vibrio cholerae, so cholera matches with bacterium. Finally, malaria. Malaria is caused by a protoctist parasite, specifically of the genus Plasmodium, so malaria matches with protoctist. So, to summarize, the correct matches are AIDS with virus, cholera with bacterium, and malaria with protoctist. Always remember, the key is to identify the type of pathogen responsible for the disease, whether it's a virus, bacterium, or protoctist. Next, we have another question which says, complete these sentences about cholera by writing words in the gaps. When the cholera pathogen is inside the small intestine, it produces a toxin. This results in the secretion of chloride ions into the small intestine. The water potential inside the small intestine will decrease or become lower, causing water to move into the intestine. Also note that, water moves by osmosis, from an area of higher water potential that is either from blood or intestinal cells to an area of lower water potential, which is to the lumen of the intestine. Now let's move on to the next question. This one is a calculation-based question. It says, in 2017, the World Health Organization estimated that 95,000 people die from cholera each year. The World Health Organization set up a project to reduce this number of deaths by 95% by the year 2030. It asks, if the project is successful, calculate the maximum number of cholera deaths in 2030. Now, first we need to understand what the question is asking. If deaths are reduced by 95%, that means only 5% of the original number will remain. So, in step 1, we calculate the remaining percentage. 100% minus 95% gives us 5%. Next, we calculate how many deaths correspond to 5% of the original estimate. The original number of deaths is 95,000, so we multiply 95,000 by 0.05. When we do that calculation, we get 4,750. So, if the project is successful, the maximum number of cholera deaths in 2030 would be 4,750. Always remember in these questions to first find the remaining percentage and then apply it to the original value. That's the easiest and safest way to solve this type of calculation question in the exam. Then, the third part of this question says, the World Health Organization recommends vaccines to protect people from cholera. Describe how vaccination can make people immune to a disease. Now, this is a 4-mark question, which means we need to explain the process step by step, not just write one short sentence. So, we will write. In vaccination, a weakened or harmless form of a pathogen is introduced into the body, which stimulates lymphocytes to produce antibodies. This process also leads to the formation of memory cells. If the same pathogen later enters the body, these memory cells enable rapid production of antibodies, providing quick and effective protection. So, in this way, vaccination makes a person immune by preparing the immune system in advance and preventing the disease from developing. In the next question, we are given a table. Table 4.1 presents information about a vaccine used by the World Health Organization to protect people against cholera. So first, let's understand what this table is showing us. The table outlines 3 main aspects of the vaccine. The first aspect is doses needed. The vaccination schedule consists of 2 doses administered 1 to 6 weeks apart. The second aspect is how the vaccine is taken. The vaccine is taken orally by mixing it into a glass of water. And, the third aspect is the storage temperature for the vaccine. The vaccine must be stored at a temperature between 2 to 8 degrees Celsius. Now, based on this table, we are asked to think about the practical difficulties of using this vaccine in different parts of the world. Now, the question asks, discuss the practical problems of using this vaccine in different parts of the world. So for this, we will simply write, vaccination may face practical difficulties because it can be hard to encourage or find people to receive the second dose. In some areas, a safe water supply may not be available, and vaccines may require electricity and refrigeration, which may be difficult due to limited access to power or refrigerators. Then we have the next question here. In this question, it says, Staphylococcus aureus is a pathogen which infects humans. It can be destroyed by antibodies and antibiotics. We are also told that the cells of this bacterium are spherical and about 1 micrometer in diameter. Along with this information, we are given a diagram. Figure 3.1 shows a diagram of an Staphylococcus aureus cell. Now, if you look at the diagram, you can see the different parts of the bacterial cell are labeled. The labeled parts are A, B, and the capsule of the bacterium. The first thing the question asks is to name parts A and B. From the diagram, we identify that A is a ribosome and B is the plasmid. Next, we move on to the following part. It says, state 2 other components of this bacterial cell that can also be found in animal and plant cells. So here, we write cell membrane as one answer, and DNA, the genetic material, as the second answer, because both of these structures are present in bacterial cells as well as in animal and plant cells. Coming on to the next part of this question. It says, antibodies can destroy bacterial cells without destroying human cells. Explain how this is possible. So for this explanation, we write, antibodies can destroy bacterial cells without destroying human cells because bacteria have specific antigens, which are proteins present on their cell surface. These antigens are recognized as foreign by the immune system. Antibodies produced are specific to these bacterial antigens, so they bind only to the bacteria and not to human cells, which do not have these antigens. Now let's move on to the next question, Part B. The question says, there are different types or strains of S. aureus cells and some can resist antibiotics better than others. A disc diffusion test is carried out to investigate the resistance of 4 strains of S. aureus K, L, M and N to different antibiotics. The 4 strains are first grown to cover the surface of separate agar plates. Then discs containing 6 different antibiotics 1, 2, 3, 4, 5 and 6 are placed on the agar plates. Figure 3.2 shows the results of the investigation after several days. Any clear zones indicate where S. aureus has died. So, what's happening here is that different strains of the same bacterium are being tested against different antibiotics, using agar plates and antibiotic discs. The clear zones show where the bacteria have been killed by the antibiotic. Now it asks, suggest why this is called a disc diffusion test. So we will simply write, it is called a disc diffusion test because the antibiotic diffuses out from the disc into the agar where the bacteria are growing. You don't need to worry too much, as this concept will be studied in detail in the upcoming chapters. For now, we only need to understand how it is referred to here. In the next part of the question, students are asked to describe what these results show about the effectiveness of antibiotics 1 and 5. For antibiotic 1, the correct description is, antibiotic 1 is only effective against strain L, as a clear zone is seen only around strain L and not around the other strains. And, for antibiotic 5, the correct description is, antibiotic 5 is effective against all 4 strains because clear zones are present around all 4 agar plates. Next it says, over time the percentage of each strain in the S. aureus population will change. Now it asks, suggest which strain, K, L, M or N, in figure 3.2 is likely to become the largest percentage of the population. So, the correct answer is strain N, because it shows the greatest resistance to antibiotics and is therefore more likely to survive and reproduce. And then, the final part of the question asks us to explain why the strain named in the first part of C is likely to become the largest percentage of the population over time. So, now we will write explanation for our answer above, which is, there is variation within the population, and strain N has the greatest resistance to antibiotics. This means it is best adapted to the environment, so it is more likely to survive and reproduce. As a result, the beneficial alleles responsible for resistance are passed on to the next generation, and through natural selection the proportion of strain N increases over time. Now coming on to the paper 1 questions. This question says, what is a method used to control the malarial vector? So, the correct answer here is C, insecticide. This is because an insecticide is a chemical substance used to kill insects. The malarial vector is the mosquito, which is an insect responsible for transmitting malaria from one person to another. Therefore, using insecticides to reduce or kill mosquito populations is an effective way to control the spread of malaria. The other options are incorrect because antibiotics and antibodies are used to treat infections inside the human body, not to control vectors, and herbicides are chemicals used to kill unwanted plants, not insects. Next question asks, which disease can be treated by using antibiotics? So, the correct answer here is A, cholera. Cholera is caused by a bacterium, Vibrio cholerae, and antibiotics are effective against bacterial infections, so cholera can be treated using antibiotics. The other options are not correct because HIV is caused by a virus, malaria is caused by a parasite, and scurvy is a deficiency disease caused by a lack of vitamin C. Since antibiotics only work against bacteria, they are not used to treat these conditions. Moving further, which row shows the type of pathogen that causes malaria and its method of transmission? The correct answer is A. As malaria is caused by a protozoan parasite called Plasmodium, which is neither a virus nor a bacterium. It is transmitted to humans by an insect, specifically the female Anopheles mosquito, which acts as a vector. Therefore, the row that correctly matches both the type of pathogen and the method of transmission is protozoan, insect. Next question says, a disease is caused by a bacterium that infects the small intestine, producing a toxin. The toxin causes osmotic movement of water from the cells into the small intestine, producing diarrhea and extreme dehydration. Which disease is being described here? So, the correct answer here is B, cholera. This description clearly matches cholera, which is caused by the bacterium Vibrio cholerae. The bacterium releases a toxin in the small intestine that causes water to move by osmosis into the intestine. This results in severe diarrhea and extreme dehydration, which are characteristic symptoms of cholera. Next, we move on to a question that asks students to consider the following statements about immunity. Which of the following statements regarding immunity are accurate? First, memory cells are generated through both active and passive forms of immunity. Second, antibodies are produced in response to antigens, whether they are present on the surfaces of pathogens or introduced through vaccines. Third, each type of antibody possesses a chemical structure that exactly matches the shape of the antigen it binds to. So, the correct answer is D. Only, statement 2 is correct because antibodies are produced in response to antigens found on pathogens or in vaccines. Whereas, statement 1 is incorrect because memory cells are produced in active immunity only, not in passive immunity, and statement 3 is also incorrect since each antibody has a specific shape that is complementary to the antigen it binds to and not identical.